Hello everyone. This is a presentation on research paper Transport-Free Module Binding for Sample Preparation using Microfluidic Fully Programmable Valve Arrays. This research paper is accepted and is being presented at the conference DATE 2020. So let's get started with the idea behind this work. Traditional laboratory experiments are performed using test tubes and pipettes. These experiments generally require reagents in volume on milliliter scale, which is large when compared to miniaturized version, which can perform the same experiment in nanoliter or even picoliter volumes. These experiments can be performed on small scale using miniaturized chips devices where a sequence of operations are required to realize the experiment. But these devices have a transportation constraint, which forms the backbone of this research paper. I'll discuss more on the constraint in upcoming slides. So, we propose a novel module binding algorithm for these miniaturized devices to perform the experiment by taking into account this transportation constraint. Here are some quick highlights about our paper. The miniaturized devices talked about are the FPVAs. They have reconfigurable 2D grid-like structure on which microfluidic operations like loading, mixing, etc. can be performed. To reiterate, we try to tackle the problem of transportation constraint in FPVAs, which says transportation of fluid in precise amounts is impossible in an FPVA. We develop a novel solution algorithm for module binding, NTM, aka no transport mixing, which does not require intermediate transportation. On top of this, we also propose a heuristic algorithm, HDA, that achieves the same experiment in much lower time. And finally, we report in our results how our algorithm performed the experiment in optimal chip area with minimum assay completion time. Here, let us introduce some of the terms that are readily used throughout this presentation. Lab on chips. Lab on chips are microfluidic biochips, which are nothing but miniaturized version of traditional biochemical lab experiments. Since they consume very little amount of reagent in picoliter range, they offer low cost point of care diagnosis. Other use cases involve their usage in sample preparation, genomics, drug discovery, etc. FPVA, acronym for fully programmable valve arrays. It is a microfluidic chip. An FPVA is a two-dimensional array of fluid chamber. Each of these is surrounded by up to 4 microvalves. So this is how an FPVA looks like, a two-dimensional array of fluid chamber. Each of these chambers are marked as white in the image, and each of these chambers is surrounded by up to 4 microvalves, which are represented as green in the image. A reagent basically flows into the input port through the chambers with open walls and passes out through the output port. Loading and mixing operations are illustrated in this figure. Fluid A in red is loaded in these cells along with loading of fluid B in orange in figure A and B respectively. As in figure C, when the walls engaged in mix operation when actuated sequentially for a certain period of time result in a uniform mixture marked as orange in figure D. Moving ahead, let's understand what exactly is this fluid transportation constraint and why is it important to consider. Consider a scenario where we need to transport a fluid from source cell to target cell as in figure A. For this, we need to first create a path for the fluid medium from input to output through source and target cells. This path is represented via open walls marked as yellow in figure B. So when fluid is moved across the orthogonal boundary, the flow gets segmented and air bubble get trapped. This is why intermediate transportation of precise amount of fluid is not possible on the chip. This is an important and more practical constraint to consider, since once fluid is loaded, it cannot be transported later. Keeping this in mind, we developed our module mapping algorithm to be discussed in later slides. With this motivation, we model our problem statement as follows. First, we model our FPGA. Since an FPGA resembles more like a two-dimensional grid, we model it as a two-dimensional Cartesian plane with center of grid at origin. Figure A shows a snapshot of actual FPGA chip, while figure B and C shows the analogy with Cartesian coordinates. Then modeling of mixing tree. A sample preparation is the generation of a mixture of fluid with reagent in specific ratios. This is an important and fairly common lab experiment that is performed. This experiment is represented by a sample preparation mixing tree as shown here. It is nothing but a directed acyclic graph. Here is the proposed workflow. We start with an input mixing tree, pass it through HDA, the heuristic algorithm, in case of optimization, else we bypass and directly feed it into the NTM module, which actually does the transport-free module binding of input tree onto the FPGA chip. These acronyms shall be discussed briefly in the next set of slides. Here is a major contribution, NTM, aka no transport mixing. As the name suggests, this proposed algorithm for module binding takes into account the transportation constraint. This takes a mixing tree and an FPGA grid as input and outputs the schedule and module binding of mixing tree onto the FPGA chip. The output contains the information of which reagent is to be loaded, mixed onto the chip at what time and at which location. Here is a worked out example of how mapping is done through NTM. Consider a sample preparation whose mixing tree is shown in figure A. The leaf node represents the pure reagent which are mixed in volume marked on the edges to get an intermediate mix node, the internal nodes. The goal is to reach the root node to get required concentration of reagent, which here is 24:18:12:10 for R1, R2, R3 and R4 respectively. For root node M1, we do post-order traversal of its subtrees and perform their mix operations simultaneously and adjacently. In first time cycle, M5 and M2 get completed as in figure B. Then by dependency rule, M4 needs to be finished before M3. In figure C, mix operation of M4 is carried out. We ensure that for mix operation of M3, one part of M4 and three parts of M5 are adjacent to each other as in figure D. Finally, in figure E, mix M1 is performed with completion of bioassay in 4 time steps. For more details about the algorithm, please go through the relevant section of our paper. Moving ahead, we also propose an optimization step for our module binding algorithm NTM. We call it as HDA or heuristic distribution algorithm. Different mixing trees may result in different module chip binding, and as shown in the last example, mix operation in different quadrant can be performed parallelly. This algorithm is based on the principle that two structurally different mixing trees can result in getting us the same mixing ratio. Keeping this in mind, we reorient an input mixing tree in such a fashion that we redistribute the mixing node almost equally in every subtree of the root node so that mixes can be parallelized in every quadrant, thereby reducing the bioassay completion time. As shown in figure left hand, we show an unoptimized regular execution of NTM with mixing tree as in figure A, and on the right hand side, figure D is the output from this algorithm HDA. Though both these trees look different but achieve same mixing ratio. Figure B and E compare their overall execution time in mixing time steps. The optimized tree is significantly faster than one on the left. Their footprint on chip are represented through heat map in figure C and F respectively. We highly recommend you to look at the paper for more details. Here are the results we obtained through our simulation of HDA and NTM. We take the time required for completion and the area utilized on the chip as metric of comparison between two approaches. In the shown graph, the NTM stats are shown in orange and the NTM plus HDA stats are shown in purple color. Though with 25% increase in actuation valves, we reduce time and area by 21 and 23% respectively. As you may see in figure A, the number of operations performed for same mixing tree are more for NTM plus HDA because some auxiliary mix node get introduced due to HDA. Figure B shows the clear advantage of HDA plus NTM in decreasing the bioassay completion time. So based on the result, we can say that HDA does a fairly good job in reducing the time and chip area for executing a bioassay on FPVA. We now come to the conclusion of our presentation with the following remarks. We have addressed the design automation problem for sample preparation on FPVA chips considering the fluid section transport problem. We've proposed a module binding for mixing tree into the FPVA and also a heuristic to reduce the assay completion time and chip area onto the FPVA. We would like to acknowledge the following sponsor for supporting this research work. Here are some of the many references that have helped us and inspired us throughout our journey of this project. Thank you, and do feel free to reach out to us.